Good morning, everybody. Are you awake? That was terrible. Are you awake? Good, because we're going to talk about research. No, we're excited. Hopefully you all made good choices last night. Yeah? I actually did. Oh, Lauren did. This is a record. No, we're so happy that you all are here this morning. We had a fantastic first day, and I'm really excited for today. It's going to be a long day. It's just long time-wise. There are going to be lots of different topics, lots of opportunities, lots of important stories and reflections and information. So please, again, think of all those things that you learned yesterday in the different coping and grief and loss sessions, and utilize the different services that are available, whether that's just taking a break or doing some of the art projects, which, by the way, if you haven't seen, those art projects are really stinking cool. So today, as we get started, we're going to start in here together with research terminology, play some fun games. I promise it will be fun. And then we'll start our concurrent sessions. So just as a heads up with where things are located, this is the Meridian. So all of our group sessions are going to be in here. We have Tico Kepler is on this side, and they're concurrent. They're two rooms together. Taurus Aquarius is on the opposite side of that, right next to the art room. Then next to us, that's Zenit. And then if you go around the corner towards the restrooms, you'll see Nadir. So as you're looking for the different rooms, that's where you can find them. Take a look. If you have any questions, please let us know. As a reminder, these sessions are all being recorded. So it's impossible to go to every single session, but you will be able to see it online later. So give yourself that little bit of grace. And then this evening, we're going to have fun, right? Who likes to dance? I like the enthusiasm. You gotta wake up a little bit. For this session, starting off, please pull out your research terminology glossary. So if you left yours, try and share with a friend. This is going to be posted online, actually on our website, so it will be translatable after Congress. So just as a note for those where English isn't your first language, we will have some other options available online. This is going to be really useful for what you're going to hear at Congress. There are a lot of other research terms out there, but we developed specifically for this event in conjunction with our research committee with HDYO. So this is going to be a good tool. What's going to happen is Rachel's going to come up and she's going to start putting these different puzzle pieces together to understand how the biology works, how the biomarkers play a fact into it, what treatments are what types of study methods are being taught, those kinds of things. And then we're going to play an interactive game. So the thought is you have it in here, you're reading along as Rachel is describing it, and then you'll start to implement it. So the hope is that as you're attending these different research sessions throughout the day, you won't feel like you're hitting a brick wall of not knowing what these things mean, right? You'll have some resources available. So we hope that this helps. So now I'm going to shut up. And I'm going to invite Rachel Harding up for our research terminology presentation.
Excellent. Thank you very much, Jenna, for a very lovely introduction. And it's such a pleasure to be here. I feel very honored to have been invited to talk with you all. So as Jenna mentioned, we're going to be going over some Huntington's disease research terminology today. And this is what I like to consider HD Research 101. We're going to be going through so much stuff, all the way from the basic genetics through to different kinds of therapeutic approaches, how clinical trials work, what kind of biomarkers are we measuring, what even is a biomarker, all of this stuff. So just to say that what you'll hear in this first session this morning is going to come up again and again in lots and lots of different sessions that you may choose to attend, some of which are listed here. So don't worry if you don't understand everything in the first run or you forget something. You also have the terminology guide as well to help you along. All right. So why am I talking to you, and who am I? Yes, it's a good place to start. So I'm actually based in Toronto, but as you might be able to tell from my accent, I'm originally from the UK, and I grew up just by the White Cliffs of Dover. I did my undergraduate and PhD at the University of Oxford, and then moved to Toronto to do my postdoctoral fellowship, and then began my independent research career there as well at the university. And so I now have my own lab. I have 12 people in my lab who are entirely dedicated to Huntington's disease research. And the kind of question that we're interested in is kind of really trying to understand the Huntington protein. So this is the protein that's changed in Huntington's disease. Figure out what those changes really mean and try and find new ways that we can develop drugs for Huntington's disease. The other hat that I wear is I'm one of the editors-in-chief at HDBuzz. And so in case you don't know what HDBuzz is, we basically write about 1 to 2 articles a week right now on Huntington's disease research news. So this can cover all kinds of things, from sort of basic science publications that come out that we think are really interesting, all the way through to news on clinical trials. And it's all written by scientists and clinicians, but we do our best to put it into plain language so it's as accessible to as many people as possible, and we aim to serve the global HD community. So if you go onto our website, there's this nice tab called a glossary, which has a kind of similar information to what you'll find in your terminology. So you can cross-reference the two if you like, if you're looking for a little bit more information. But as I go through the slides today, every term that is covered within this terminology guide is going to be highlighted in yellow on the slides, so you'll know which words to look out for and which words really matter. All right, let's get into it. So my background in training is in protein biochemistry, and what that really means is that I'm really, really interested in understanding what happens in biology at the molecular level, and this is where all the good stuff is really happening, and that's what we've got to really try and understand. But just to put that into perspective of what that means is, you know, I'm actually almost 2 meters tall, my brain is 15 centimeters across, but if we go all the way down to the next level down, which is the cells or a nerve cell, we're now at 20 micrometers. But if we're thinking about individual molecules within those cells, they're 5 nanometers. So what does that really mean? Well, what that means is if I scaled this tiny, tiny little nerve cell to be as long as I am tall, a protein molecule would still only be as big as the thickness of my fingernail. So these are tiny, tiny little things that we're thinking about, but they're really important to what happens in HD and other diseases. So every single cell in our body has a copy of all of our DNA. This is called our genome. And the genome is organized into these nice little packages that are called chromosomes. And on chromosome 4, this is where you will find the Huntington or HTT gene. And our DNA is basically the instructions for, sorry? Okay, sorry. So our DNA is basically the genetic instructions for how to make all of the really important molecules that do all the stuff in biology. And so our DNA is our first copy, but our body wants to really look after our DNA. And so what it does is it makes a copy of it, which is called messenger RNA. And in the case of Huntington's, this means that our Huntington gene in our DNA would be turned into this message molecule, which is then turned into our Huntington protein. And so we can think of this a bit like doing some cooking in the kitchen, where our DNA is a bit like the cookbook, our RNA is the individual recipe, and then the protein at the end is whatever you make, whether that's a cupcake or something else. And so you probably already know lots of proteins that do the stuff in cells. So these are things like keratin, which is important for your hair and nails, hormones, which are things like insulin, all the enzymes that digest your food, and sort of antibodies, which you'll make if you have a vaccine or something. But what does the Huntington protein do? Well, Huntington is really interesting. That's why I'm very excited to study it. And we like to think of it as almost like a Swiss Army knife in the cell. It does all of these different jobs, and it's involved in lots and lots of different processes. And this is why, there's a lot of research to try and understand what goes wrong in HD. So what actually causes HD? Some of you are probably familiar with this, but we all have this Huntington gene, or the HTT gene, and at the very, very beginning of the gene, there are these repeating DNA letters, CAG, 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 over and over again, And anywhere from sort of 10 to 26 is very typical. But in folks who have HD, you'll have an expanded form of this gene. And what this means, instead of having 10 to 26 CAGs, you'll probably have anywhere from 40 and above. And these CAGs are encoded right at the very, very beginning of the Huntington gene in this region that scientists call exon 1. So you'll hear about that a lot in different talks that will be happening at this Congress. And so what does this mean? So you've got this change at the genetic level, but like how does that transpire into being Huntington's disease? So essentially what happens is, because our DNA encodes the protein, if you have a regular form of the unexpanded Huntington gene, you'll make the regular or wild-type Huntington protein, and you'll have healthy nerve cells and things should be okay. But if you have HD, you'll have an expanded form of this Huntington gene, which means that you also will then make an expanded protein. Because remember, your DNA is basically the recipe for making these proteins. And we also sometimes call this mutant Huntingtin or MHTT. And when we do this, we'll often get sick nerve cells, and then this is where we see the signs and symptoms of HD. And so this is something that you might come across a lot. This is one of the first things that will come up if you Google Huntington's disease. Is one of the most obvious ways to characterize this disease is the loss of brain cells or atrophy within the brain of very specific regions. And so there's this part of the brain that's called the striatum, which is kind of pointed out here, which kind of, we lose nerve cells over time. And that's why Huntington's is called a degenerative disease, because we're losing these cells, we're degenerating. And the striatum is made up of these two other parts called the putamen and the caudate, and those get smaller over time in people with HD. The region of the brain that gets bigger is actually this little gap that you can see here. This is actually called the ventricles, and you'll see that those get larger. And so often these kind of measurements, these changes in brain structure, are what we'll look at in different clinical trials. Okay, so Huntington's disease is inherited from our parents, and everyone will get one copy of the Huntington gene from their mum and one from their dad. And what will happen most of the time is that most people will make a healthy form of the Huntington protein and then a disease form. So most people only have one copy, which is the expanded form of Huntington. And what this means is that if we go back to our cupcake analogy, it means that you'll make these two different versions of the Huntington protein, one of which works very well and is pretty normal, and the other one which is expanded. And we can think of this as like some really nice, beautifully made cupcakes, and others which turn out not so great. And then what happens from there is we know that when this expanded form of Huntington is made, we have all of these symptoms, which most people in this room will be very familiar with. And these cover sort of physical, emotional, and cognitive symptoms. They can be quite varied between different people, but this is kind of a nice summary from the HDA. So what we need is, we need drugs, right? So that's what we're all interested in. That's what most researchers in the field of HD are interested in discovering. There's different kinds of drugs that we need, and we need all of these. So we need stuff that's going to help treat symptoms. We need things that are going to slow down the onset or progression of symptoms. So that means that things will just happen a bit more slowly. We can delay the disease. But in an ideal world, at some stage, we will have drugs that will either stop HD in its tracks or even reverse symptoms and make things better again. Avenues that different people are exploring. But before we get to a drug, we have to go through clinical trials. So I'm going to go over kind of how this works. People like myself, Lauren Byrne, lots of other researchers who are in the room right now, we're all kind of in this preclinical stage. This means that we're at a bench doing wet lab research, we're doing experiments, figuring things out, and trying to find new ways that we can develop medicines for Huntington's disease. And then we'll move into the clinic. And there are different phases of trials. You might have heard of some of these, and you hear, oh, it's a phase 1 or a phase 2A, or other kind of combinations of phases. But essentially, there's typically 3 phases, 1, 2, and 3. These start in, generally as you go through the phases, you start off with a really small group of people, and it gets bigger over time. And you begin just checking out safety. That's the most important thing in a clinical trial. Then in phase 2, you want to check, is your drug that you've made, is it actually doing the thing that you wanted it to? And when you're in this larger number of people, is it still safe? And then you get to phase 3, which is like, is your drug actually changing the disease? This is what we call disease modifying. And so the way that these trials are designed is at the very beginning, the scientists have to set out exactly what they're going to do. So how are they going to treat people? What are they going to measure? How is it going to go? What kind of dosing are they going to do, and so forth. Then they're going to recruit people with inclusion and exclusion criteria. So this might be how old they are, how progressed they are in disease. And then we go into, exactly what drugs or interventions you're going to have, what measurements you'll make. And at the very end, you're going to see if your trial meets something called an endpoint. So this is where you set out at the very beginning of your trial, like what you would deem to be a success. And did you meet that or not? So most trials, we're not just waiting till the very end of just giving people drugs the whole way through a trial and then just seeing what happens at the end. We want to make measurements the whole way through. So these are called longitudinal trials. And so here's an example of one of the first trials that looked at tominersen, which at the time was HDTRX. And what they're looking at is this drug was designed to reduce the amount of this mutant Huntington MHTT, and we're looking at different time points after or when people were given the drug. So we want to see how is the drug working over time. The most important thing is safety. So any clinical trial, the most important thing we want to look at is the safety of the drug and are people okay? Are there any side effects that we don't want? These are called adverse events. And although we'll look at this at the very end of the trial as well, there's another group of people who are kind of like health and safety inspectors in a clinical trial. And so they are called the Data Safety Monitoring Board. And they'll be looking at the data as it's being generated through the trial to be like, is this drug working? Is it doing what we think it's meant to be doing? And is it safe? Are there any red flags we need to know about? Because the scientists who actually design the trial don't actually get to look at the data until the very end often. And the way we want to know is a drug working is that we'll often test against a placebo. So most people will be familiar with this. This is kind of like having a dummy version of the drug or like a sugar pill. So some people will get the drug, some people will get the sugar pill, and often you don't actually know who gets which. So there are this is called the way that the study is blinded. So normally the people who are taking the drug will not know which one they get. And then the researchers who are organizing the trial often don't know either. And actually the people analyzing the data sometimes don't know. And they've got to try and find, is there something real? Can we actually see a difference between these? And these different groups are called the sort of control group and the treatment group. And so what we want to see is, do the people in the treatment group get better compared to the people in the control group? But you can say it's probably a little bit unfair for people who go through clinical trials and then all they've been given the whole time is a sugar pill. And so often what will happen is this thing called an open label extension, which means that people who were in the control group will actually get access to the drug in a kind of at the end of the trial, and they'll be continued to be monitored. So when a trial ends, we already know what we're going to look for. That's detailed in the study protocol. And so basically what we want to do is see, did we meet the things that we wanted to? Did we hit the criteria that we set out to know if a drug would work or not? We can also look for other trends in the data. Sometimes you'll get some really interesting things come up in clinical trial data that you kind of want to look into and dive into. And this can form part of what scientists call a post hoc analysis. But even when a trial has ended and even if it meets all its endpoints and everyone's really happy and the FDA approves it and then everyone starts getting treated with this drug, things don't end there. We often have what we call phase 4 of clinical trials, which is or post-market surveillance. This is where people in the real world who are given the drug are continued to be monitored by their doctors and other people just to make sure now we're giving the drug to even more people. Is it still safe? Is it still working as we want? The good news for diseases like Huntington's, because it is considered like very serious and rare, there are all kinds of ways that the different regulators will have to try and speed up how quickly can we get drugs that work into the hands of people who need them most. And there's all these kind of accelerated programs or early access programs, which means that we're just sort of hitting the gas pedal as much as possible to move things forward. That said, it is still really slow. This whole process from sort of very, very early discovery the whole way through can be 5, 10, 20 years. And there's something to keep in mind when you're reading like different scientific papers and people say, oh, this could be a good treatment for Huntington's. We have to just kind of remember that there's still a long road to go. And this path is really important because we want to make sure that we have drugs that really work and are safe for people. The other thing to remember as well is that, as much as we do our best, scientists are trying their hardest to find the best kind of therapies, the best kind of drugs that they want to try and treat HD with. In general, most trials don't pan out the way that we want, and treating diseases in the brain is very, very difficult. So we'll start off with all of these different ideas and all of these things at the beginning, but actually the number of drugs that are approved by different regulatory bodies through the world is actually quite small. So we kind of just have to manage our expectations sometimes. But the good news is, the more stuff that comes in, the more likely we are to have something really good come out the other end. And that's exactly what's happening in Huntington's disease right now. So on here, I've got logos of companies that have either they're currently working on HD or that they've expressed an interest in working on HD, which is super, super awesome. And you can see there are all of these companies based all over the world who are working really, really hard towards the different kinds of drugs that we want. And the other good news is that people are investing in this. Drug discovery is a very expensive process, and we can't do this without money. And you can see from this graph here that the investment in the past and also in the predicted future is going up a lot for Huntington's disease. All right. Okay, so what are people actually doing? Well, we have different types of therapies that we want to think about. So remember earlier we talked about stuff that affects symptoms, but what I really want to focus on today is what we call disease modifying. So things that slow, halt, or reverse the course of disease. And this slide is from my colleague Sarah Hernandez. She put together this really nice graphic so we can see all the different stuff that people are trying. And she did an even better job of just annotating this with all the companies so you can see exactly who's doing what. And I think this is so exciting. When I started working in the HD field and go to therapeutics conferences, we were testing vitamins in people. And now we have all of this amazing stuff with big companies. This is like Pfizer. These are just enormous companies with huge resources, all invested in these therapeutics, which is amazing. So one of the main ones that you've probably heard about is called Huntington lowering. So what does that mean? So let's go back to this. So we know our DNA encodes the Huntington protein. We make this copy of it that's called the RNA, and then we have our expanded or mutated Huntington protein. And what nearly all of these therapies do is they tackle this kind of middleman, the RNA, to get rid of the RNA or do something to it, which means that you end up with less of the expanded Huntington protein. And there are all kinds of different ways you can do this. So you might remember we said earlier about how everyone with HD will make a healthy form of the protein and a disease form in general. And so some companies take what we call our total Huntington lowering approach. So what this means is they're getting rid of both forms of the protein to a certain level. Others have, kind of very clever genetic tricks, which means that they only get rid of the disease form of the protein. And this is what scientists call allele selective Huntington lowering. And there are all kinds of different ways that we can deliver these drugs. And so we have things like antisense oligonucleotides, RNAi. These are drugs that are typically given by sort of spinal injection. We have other drugs that are taken by mouth, or they're either in tablet or syrup form. These are called splicing modulators. And then there's another form, which is actually a gene therapy, which is this virus-based drug, which is actually given by brain surgery. And so all of these, they work in slightly different ways, and you're probably going to hear more about this later on, and I'm going to talk about it again tomorrow. But the main message is that they interfere with the RNA molecule of Huntington, which means we end up with less protein. And the reason that we have to give them in these different formats is because they're different types of drugs. So small molecules, in the very name of them, they're small. And then we have these which are kind of medium-sized and then even bigger. And basically, the size of the drug really matters because the bigger it is, the harder it is to cross the blood-brain barrier. So this is like your body has this very protective barrier around your brain. Your brain is very important, so your body wants to look after it. And essentially, really big things have a difficult time getting across. All right. So a new and exciting area of Huntington's disease drug discovery is something called somatic expansion. And if you read HDBuzz, you would have seen we have a ton of articles on this right now because it's a really, really hot topic and there's all kinds of really cool stuff happening. But this is still quite early phase, like none of this is actually in the clinic yet. And essentially what this is, somatic expansion is, we all inherit our Huntington gene, but in people with HD, most of their cells will have exactly the same CAG number as they get older and older. But in some brain cells and in some other cells, actually the CAG number can get bigger over time. And we've known about this for a really long time, but it's been difficult to kind of connect the dots about what's happening. There we go. And what this means is that in your brain, this is a really nice graphic from what used to be triplet, you can see that you'll start off with all of your cells have the same CAG number, and as you get older and older, some of them will get really, really big. And we actually were able to figure out maybe what was causing this because of people like you in this room who donated different samples so we could do genetic studies on hundreds and hundreds of people, and these are called genome-wide association studies. This is where we're looking for what's going on and why symptoms for Huntington's disease can come earlier or later in life. And you might hear about this when you speak to different people in this room, that even people with the same CAG number, some of them will get disease 10, 20 years apart. And so why is that? And that's what this study looked to try and find out. And what it found out was there are all these other genetic modifiers which change when symptoms can begin in HD. Oh, there we go. And it turns out that many of these are actually things that affect somatic expansion. So this kind of ties things really nicely together, where symptoms are changed because of somatic expansion, which is influenced by some of these other genetic factors. And so there's a whole load of companies who are trying to mimic this. Essentially, what's happened in nature is that nature has already provided us with answers about how we can slow down Huntington's disease. And so what companies are now trying to do is say, well, this person has this genetic change, which meant they got Huntington's disease later. How can we replicate that with a drug or a therapy? We also have people doing some really quite radical and fun things with cell replacement therapies. This is where you can take skin cells from someone, turn them back into stem cells, and then turn them into all kinds of different cells and implant them back in the brain. And the idea is that you really want to help restore connections because of the nerve cells that are lost and reconnect these synapses. There are other people doing even more crazy things, and it's really, really interesting science, where actually you can reprogram cells in your brain that are not nerve cells and make them become nerve cells. So what you can actually do is turn these cells back into nerves and help rebuild connections in the brain as well. We also have people looking into other kinds of ways to make therapies. There are people who are looking at trying to restore how different cargoes are carried along nerve cells, which is something that goes a bit wrong in Huntington's disease. And then we also have people looking at sigma-1 receptor activation. And I'll go into a lot of these in more detail tomorrow. Another thing that you've probably heard about, which is this big kind of advent in sort of gene therapies, is CRISPR. And CRISPR is this amazing toolkit which allows us to make very, very precise changes at the DNA level. We always liken it at HDBuzz to having almost like genetic scissors. So you can kind of chop up the DNA, change it to what you want, and put things back together again. And there's nothing in the clinic yet for Huntington's disease, but something that's really nice is that all the papers that are published on CRISPR and HD, and you can see this is when CRISPR was first sort of published, and over time, the interest is really, really growing, which is super, super awesome. And there are labs all around the world thinking of different ways we could use this. So this isn't just about, you could use it, for example, to actually reduce the CAG number, to lower Huntington protein, or maybe influence a genetic modifier. And yeah, so lots and lots of options. It's very versatile. Okay, so now I'm going to switch gears a little bit. These were all things which would actually change disease course and stuff that people are investigating. Another really important kind of study that we'll have in HD and other diseases is what we call non-interventional studies. So these are studies where you're not given a drug, but different kinds of data is recorded. And so these can include things like registries. So this is Join HD, which it basically allows sort of registration of juvenile Huntington's disease cases. And this kind of stuff is really important, especially for diseases which are very rare, because we just need to know how many people actually are out there who have this. Like, having that data is really powerful, and it's really important for sort of lobbying and making sure we have sufficient resources. And then we have things like observational studies. And so the one I'm highlighting here is the HD young adult study. And this particular study is basically taking lots and lots of different measurements of people who are at the very earliest, they're gene positive for Huntington's disease, and they may or may not have some of the very earliest symptoms. And it's just trying to find out, like, what does that look like? We've spent a lot of time looking at Huntington's at sort of very end, but what we really want to know is how it looks at the beginning, because this will allow us to do clinical trials in younger people. And then obviously I have to highlight surveys, and if you haven't already, I highly recommend that you take part in the HDYO survey series. All of the information is on the website. Okay, probably one of the most important observational studies in HD is something called Enroll HD. And if you're over 18, you are able to be enrolled into this study. You don't actually have to know your genetic status. But, there's different bits and pieces that are collected over time. This is like one of the largest studies in the world on Huntington's, with over 20,000 participants. And this means that we can get these really huge and powerful data sets and make all kinds of interesting discoveries. And I would highly recommend, I know there's people here from all over the place, but there are lots of really small local studies that happen at different HD clinics. They might be to do with, how exercise might impact your disease, or medicine combinations. There are other stuff like HD Clarity, which has sites around the world. This is collecting spinal fluid from people or CSF from people to make different measurements and make different correlations. All of this stuff is really, really important. So even though you're not receiving a drug, actually this kind of data is just incredibly powerful. And this is where we can make discoveries about things like biomarkers. So biomarkers are basically these things that we can measure that track with the disease over time. And so, in people with HD, what we've already talked about is this could be brain imaging, so we know from an MRI exactly which parts of the brain are changing. It can be taking CSF samples, as we said, from the HD Clarity study, and then just sort of seeing what's actually happening in the spinal fluid of people with HD, and also looking at blood and plasma samples. Something that's a really important biomarker, and you're going to hear a lot about this, is this protein that's called neurofilament light, or NFL. And essentially, it's a really, really good surrogate biomarker for understanding brain health. And it's very simple. I like this as someone who's not a clinician, to understand, where basically more NFL means that your brain is not doing so good. And so it's already a biomarker that's used in all sorts of other brain diseases. And so it's now being used more and more in HD. All right, so what other measurements can we make? So there are other things that we call clinical outcomes, and these are ways that we can measure signs and symptoms of Huntington's disease. And there are all kinds of different measurements and scores that people can use towards this. Biomarkers are also part of this, but we have all of these scoring systems which are about the way people are able to live their lives, like their cognition, their movement symptoms, and so forth. And some of the most commonly used ones are this total motor score, total functional capacity, and single-digit modalities test, which together form this UHDRS rating scale. And so this, you'll see, this is commonly reported in clinical trials for different outcomes to see how do these measures change if we give someone a drug. And together, all of this kind of research to understand how disease is moving forward has been really, really critical to make what we call a staging system for Huntington's disease. So we'll be very familiar with staging systems in other diseases. So if you know someone with cancer, they will tell you, if they're stage 1, 2, 3, 4, and so forth. And this basically tells you how far along your cancer has progressed. And we had all kinds of different ways of doing this previously for people with HD, but they all kind of overlapped a little bit differently, and it was a bit complicated. And so a whole group of scientists from around the world came together to try and figure out, well, how can we figure this out and make something that works for everyone? And so we have these stages from 0, 1, 2, and 3, where 0 is just that you have a positive test for Huntington's disease at a genetic level. And then 1 is where you start to see some subtle changes in biomarkers. 2 is where you start to have clinical signs of Huntington's disease. And then 3 is where you have more progressed symptoms. And this kind of stuff can seem quite boring and administrative, but it's actually really, really important, because if we're going to do a clinical trial where we start to treat people at this stage, how do we know that the drug's working and things are getting better? And so what we can do is look to see, well, how long does it take them to go from 0 to 1? Does the drug make things move forward more slowly? And this is actually really helpful for treating people at earlier stages of HD, which is what we really want to do. Okay. So that kind of wraps up a lot of what we're going to talk about today. As I mentioned, we kind of whizzed through 10,001 things here. So I really, it's a lot of information to take in, but I really recommend that if anything particularly caught your eye, there are all of these other talks, which is really, really exciting to see, and it's great to have so many people from industry and academia here to come and talk to you about science. And I was particularly intrigued by this one, because there's some very nice titles here. Taking a Whack at Mutant Huntington, which I think is something we all want to do. Okay. And so just a reminder that tomorrow at 9 o'clock, we're going to have an HD Buzz research breakdown. And have your metaphorical golden tickets, which is a slight preview of the theme of the talk that will be coming tomorrow. I just want to acknowledge that many of these slides came from Sarah, Tam, and Leora, who are part of the HD Buzz team. And HD Buzz is supported by all of these different folks, which we're very, very grateful for. And most importantly, thank you. Thank you for being here. This is like being at a conference like this. This is advocacy. Taking agency in your choices and learning about this disease is really, really important. And none of this research would be possible without HD families. So thank you very much.
It's a lot of information, but it's great to see how it all works together. And we don't expect you all to be experts right now, but I think this opens the door for more questions, because we're always continuing to learn more and more each day about this. So now what we want to do is play a little game called Jeopardy. Has anybody heard of Jeopardy before? Some people haven't. I know it's a little bit of a different game show, but Jeopardy is a game where it's kind of a trivia game, and you are asked clues to try to answer what is the question that they're asking. So if I was to say, this covers the tables in this room today, you would answer, what is a tablecloth? Would be the actual answer to that. And so we're going to be asking or giving you clues based off of what you heard today and what is in your research terminology that will give you the question that we are asking. It sounds more complicated than it is, but the good news is you're working together in your table groups. And it's going to be fun. There is no winner or loser with this. This is just trying to take the information and start applying it. Does that make sense? So what we're going to do is Rachel is going to be choosing which questions that we ask based off of the levels, and then we'll ask it to you all. And if your table feels like they have the answer, raise your hand. And then we'll come to you to see if it's correct. Yeah, scientists, you need to disperse. Go sit at other tables. Feel free to move tables right now if you feel like you need to. But it's going to be fun. Yeah. are in the genes. Get focused. Types and methods. Measuring up. Mark it up. And Clinical Trials 101. All of these titles are hints with where you will find the answers in your research terminology. So if the question or if the clue is in measuring up, that'll be the different measurements and outcomes. If you're looking at get focused, that's what's going on in science right now with the focus on what they're doing in the different clinical trials and studies. Mark it up as biomarkers, okay? So that's kind of your cheat sheet for that. All right, Rachel, let's do this.
Okay, as a British person who's only ever watched one episode of Jeopardy in my entire life, I do apologize in advance if I don't do this right. I think I meant to say, can I do Clinical Trials 101 for $200? Is that right?
Yes.
Okay, so here we go. Administering a pre-specified amount of a therapy to determine if it's safe and effective.
So what is the answer? Look at your definitions in clinical trials section. Oh, we have Sadie, you were first. Tegan. Yeah. What is dosing?
Wait.
Hey!
Very good. We're off to a great start. Oh. All right.
Can you should be able to click on it. Oh, that's not working. It should just go to the next one. There we go. All right.
Okay, next, why don't we do get focused for $400? Okay, getting selective. This approach only lowers the mutant Huntington protein.
You already went. Yes, right up here. What is allele selective Huntington lowering? Yay!
Absolutely. Very well done.
Right.
Okay, let's go for types and methods for $600. Okay, HDYO launched one of these in 2023 to collect information from individuals about their experiences. Oh, over here.
I'm going, oh, do we have one back there? Oh, Claire, you work at HDYO. That's a cheater. No, I'm disqualifying.
Okay, disqualified
Right over here.
immediately. No further participation.
What is registries?
That's a close one, not registries, but I'm going to give you another chance. Yep. Oh, whoops. What? Yes. What is?
Were you going to say what was the survey?
What is a survey?
Right.
Yes.
Excellent. Okay, let's do measuring up for $200. This is a way to measure symptoms, overall health, ability to function, quality of life, or survival outcomes in a research study. And I have a lady right here who's ready to give me an answer.
What are clinical outcomes?
Oh. Yes, excellent.
Woo-hoo.
Very well done. Okay, let's next go for in the genes for $800. Okay, the original chat function. This carries instructions to tiny machines inside the cell, which read the message and build protein.
Gloria was first. I saw the hand.
What is messenger RNA?
What is messenger RNA? There we go.
Excellent.
Did you like my Facebook reference there?
Okay, we haven't done mark it up yet. Let's do mark it up for $1,000.
Ooh. 1,000. Mark it up for 1,000.
Okay. 2 of these fluid-filled cavities are commonly measured in HD. As HD progresses or in the presence of inflammation, these expand. Over here.
What are ventricles?
You are correct.
Woohoo!
Excellent job. Okay, all right. Let's do some more gene stuff. In the genes for $400. Wild thing. This protein plays a role in brain development and neuronal survival. It is the Huntington, it's unaffected Huntington in HD.
What is the wild type Huntington?
Absolutely. Dun, dun, dun. All right, let's do another mark it up for $400. Tip your cap. This commonly used measure may help predict the progression of HD.
What is a CAP score?
I think you're right.
Rachel, you should know this.
I should really know all the answers, but I am very jet lagged, and yeah.
It's not going.
I was also going for the dramatic reveal. There we go. Okay. Would someone like to select one? No volunteers. Okay.
I want to do one.
Yeah, let's go for
And
it.
I'm going to read this one because it's a funny one. I just like it. This is what I did in the middle of the night one night. Let's do clinical trials for 400. Goal! This measures whether a treatment meets its intended goal.
What are endpoints or testing endpoints?
It could be both.
I think it could be both.
Yeah, I appreciate that.
Yeah. Yeah, you get the $400.
Be honest.
Megan, do you want to pick one?
Yes. Okay, how about clinical trials for 800?
Clinical trials for $800. Okay, this phase of clinical trials is a medium-sized trial, 50 to 200 people, testing both safety and efficacy.
What is a phase 2 study?
What's it? Sorry.
A phase 2.
Yep, absolutely. What is a phase 2 study? Okay, let's get focused for $1,000. Okay, this therapy involves inserting a gene or genetic material into the genetic code of a target cell type to manipulate the proteins those cells produce.
What is gene therapy?
Absolutely. $1,000. All right, let's do measuring up for $800. This research tool is used to classify disease progression in HD. Oh, getting my steps in.
It's the Composite Unified Huntington's Disease Rating Scale. You say what is? Oh, what is? My bad.
Let's have a look. Yeah, so that's a way that we can measure disease and sort of see how far people are sort of, they get a score as like things change through disease progression. But the integrating staging system is what kind of sets people at different sort of milestones through Huntington's disease. Yeah. Okay, let's do types and methods for $200. Okay. This is a procedure used to collect cerebrospinal fluid, CSF, from the lumbar region or spinal canal. Oh.
What is lumbar puncture?
You are correct. Okay, would anyone else like to choose one? Oh, no, any takers? Obviously, Lauren Byrne wants to choose one.
Thanks, Rachel. I'll go for types of method for $1,000.
Types of methods for $1,000. Okay, size matters. Often taken orally, this is a type of drug that enters cells easily because it's so small.
Oh, right here.
Oh,
What is a small molecular drug?
dun dun dun. Hey! Very good. Okay, oh, we're about halfway through. This is great. Okay, let's mark it up for $800. This is a research tool for classifying disease progression. Oh, okay. Yeah.
Oh, I get it. Okay. What is integrated staging system?
That may be an error on my point. I think we had two of those.
But you got it correct, and you have $800. Well, repetition is the best way to learn these things.
Yeah.
This is how I learn everything anyway.
It was meant to be.
Exactly. It was all planned. Okay, let's do get focused for $600. You're not catching a cold. This harmless virus can be used in gene therapy to deliver DNA instructions to cells so they can make proteins. You already had your hand up before halfway through. That's amazing.
What is adeno-associated virus?
Absolutely. Yes. Well done. $600. Okay, I'm going to come more towards the back of the room. It's been very quiet in this corner. All right. With someone on this table, would you like to pick one?
Measuring up for $400.
Measuring up for $400. Okay, this analysis compares the risks of a situation and its benefits. I think the clue's in the question. Is anyone with that? Oh, here we go. You have a very gentle hand up over here.
What is risk-benefit analysis?
I think you're right. $400. Okay. What about this table? Someone over here want to choose one? Yeah, you will.
Rachel, I'll take in the genes for $1,000, please.
In the genes for $1,000. Okay, this process changes the CAG number in some cells over time. Oh.
Sorry. What is somatic instability?
Absolutely. Somatic instability or expansion, yes. So we call it instability because things can actually move in both directions, but generally things tend to expand. That's what we're interested in. Okay.
Maybe
Lauren,
back here. Anybody want to ask a question? Come on, Anna Maria.
I would go for get focus for $800.
Get focus for 800. Okay, an editing process of the messenger RNA that involves introns, leaving only the protein coding regions called exons. I didn't cover this in that much detail, but it was briefly mentioned.
Oh, right here.
Okay, I think splicing.
You are correct. What is splicing? $800.
Good job.
Well done. Awesome. Okay. I'm going to find someone in the Sabrisi lab. Shall I pick one for me?
Yes. Let's go for in the genes, $600.
In the gene, $600. The stretch of DNA at the beginning of the HD gene, which contains what three-letter repeat?
Oh, right here.
CAG.
So I think the CAG is the repeat.
Yep. What is the three?
Oh,
the CAG
oh.
repeat? Which
I thought
three
it was gonna be
letters?
X on one,
Which
so-
3 letters?
Yeah, I can't read questions, so there we are. There we go. All right.
Good job.
Very well done. I'm going to come and ask these 3 people over here to choose one.
Get focused for £200.
Get focused for $200. This is a strategy that aims to reduce the amount of mutant Huntington protein
Oh, it says in.
in cells, I would imagine, or the body.
It was covered.
Oh, I'm coming. I'll be right with you. Okay.
What is Huntington lowering?
Absolutely.
Way to go.
Important to remember, though, Huntington lowering can be total Huntington lowering, so it's not always just a mutant protein. All right, let's do mark it up for $600. Okay, along with its buddy, the putamen, this is a small region deep in the brain impacted by HD very early in disease course.
Oh, right over here.
Jenna's on it. There we go.
What is caudate?
I'm not a physiologist, but I think that's correct.
Yes.
Awesome. Okay, I'm going to ask Laura. She's right here, so I'm going to ask her for another one.
Rachel, I'll go for clinical trials 101 for $1,000.
Clinical trials for $1,000. Okay, this is an analysis that examines data after a study is complete. You can't put your hand up, Lauren. Anyone? Oh, I'll come back over here.
What is post hoc analysis?
Correct. $1,000.
We probably have time for 2 more.
2 more. Anyone wants to choose one? Okay, I'll choose one. Okay, let's do measuring up for $1,000. This single score is calculated based on 4 scales of the UHDRS and is sensitive to measuring progression of HD.
Erica's right behind. Oh, yeah.
Oh, hang on. Oh, yeah, I'll come back to you. Right.
What is composite unified Huntington disease rating scale?
Absolutely. CUHDRS.
Good job.
Okay, we'll do one last one. Okay, let's do types of methods for $800. Okay, it's time to intervene. This type of study involves human participants to evaluate safety and effectiveness of new treatments.
Oh, right behind you.
Yes, hi, Rachel. What is clinical trials interventional studies?
You are correct for $800. Awesome.
All right, round of applause, everybody. Thank you so much for participating and working us through some of the hiccups, because that always happens, but mainly my fault. But again, what did we learn yesterday? If anything goes not all the way best, who do we blame? Just kidding. But let's go ahead and take a little bit of a break between now and your next set of sessions. So again, we're going to start those concurrent sessions. So get some water, get some coffee, get some tea, use the restroom, chat, go visit some exhibitors.